Yeah, no, it's
on.
So you already know my name. I'm here to represent Akribion Genomics, new spin-out about to be incorporated next year of the Brain Biotech Group from Zwingenberg in Hessen on the Bergstraße. And I'd like to present to you today a new class of cancer therapies. Why do we need yet another class of these cancer therapies? There's so many out there, but at the essence, cancer is a complex disease, and we need many different ways to treat it. There's not one fits all solution. There's several major approaches to do that. I want to introduce two to you today. The first is the classical cancer treatment that really focuses on inhibiting or hijacking pathways that are altered in these cancerous cells. You need a detailed mechanistic understanding of the cells, and you also need to find a very specific inhibitor or active compound that targets these pathways. And the aim is here to stop the growth of cancer cells or also to take away their basis for survival. But there's also a different way to do this, and that's called the guided toxicity. You just want to recognize these cells as different, as cancerous, and you want to get rid of them. To do that, you need an address label, a biomarker that marks them as different, and you'll also need a toxic payload in order to kill them. And that's exactly what we have developed. We've developed a new technology here called a molecular shredder that's activated by RNA-based biomarkers, and it's called GDASE. It's a three-component system. First of all, the payload. That's our GDASE enzyme. It's a nuclease, so it shreds DNA and RNA with a very high activity that actually causes cell death. But it only does so once it's activated. And the activation is based on a seeker that we need to find the right sequences. That's our guide RNA. It binds to a short stretch of RNA that is present within the cells, and upon this, activates the enzyme in order to initiate the cell death. The third component is a delivery system, and we need this in order to get this complex into the cells. We're currently working here with a protein-based approach that couples these RNPs, the proteins, to inorganic nanoparticles, but we're also developing other delivery methodologies in order to be flexible in the different tissues we can target. And this technology is already patent protected. Our main patent was granted last week, actually. It covers the sequence and the compositions of this, as well as the major application fields, and we have additional families of patents filed. So now this technology, how does it compare to other technologies out there for guided toxicity? The current gold standard, I'm clumping in two different approaches here, are ADCs, antibody-drug conjugates, or cell therapies, for example, CAR-T or NKT cells. They offer very high specificity and a very high toxicity against these cancer cells, and a lot of them are already on the market. However, they have a limited target range. For the ADCs, antibody-dependent, it's because they can only target antigens on the surface of the cells. You're limited there, while the CAR-T cells or other cellular therapies can only target aberrations, mutations that are present on a protein level. Another technology is other guided nucleases. A little bit similar to ours, they offer a very high target range. They offer very high flexibility in adapting these approaches because all you need to do is change the guide RNA. And they offer very high specificity down to a single base pair level. But what they do lack is the necessary toxicity to kill the cancer cells. And our technology covers all of these aspects. We make new targets available with the wide target range of the RNA-based approaches. We can very easily reprogram our system to target a different cancer indication by just switching out the guide RNA. And we offer lower off-target effects because of the high specificity with a better side effect profile that is expected. To show this, we are currently working in vitro stages on preclinical models. And just to illustrate this here in this experiment, we've used a co-culture of cancer cells. These are the green ones marked with the GFP, and control cells that are marked in red. And the green cells carry a biomarker that's an HPV protein called E6. And if we transfect these cells with our system, when we target the cancer-specific marker that's not present in the healthy cells, we kill over 98% of these cancer cells. And the healthy cells are completely unaffected. First of all, we want to turn this into a treatment for HPV-positive head and neck cancers. We have a strong scientific rationale there because we know the biomarkers very well. And it also has a high unmet medical need, especially on the side of better side effects. In the long run, we want to turn this into a true treatment platform. We've already identified over 20 molecular targets and keep finding more across 5 different target classes. These are the aberrations that we can target. Not limited to viral proteins, but we also have fusion proteins that we can target, alternatively spliced proteins or RNAs, point mutations, and also non-coding RNAs that are addressable with this. And with this platform potential comes a huge value potential as well. Our lead indication has a total addressable market of over $2 billion alone. And with the other indications that we're currently identifying, we're looking at a multi-billion-dollar opportunity with an IPO or exit potential of far north of $500 million. And in order to make this value come true, we've put together a strong team. Most of us are actually seasoned biotech executives from Brain Biotech, our parent company still, including former CFO, our former head of R&D, Michael, who is sitting also in the audience. The principal inventor of the technology, Paul, and Dirk, with a strong strategic partnership experience. We plan to add additional expertise, especially on the preclinical development side, and also on a business development and networking side for the US in the long run. Currently, we're establishing the foundation to actually make all of this possible, and we're raising a Series A funding round to do that. The first core aim is to actually spin off the company. Our parent entity, Brain Biotech, is still fully focused on industrial biotech applications, but we are building a fully dedicated and focused therapeutics venture. On the other milestone side, we want to define 2 preclinical development candidates and also initiate the preclinical work for those. We intend to set up an R&D grade production process and prepare for a GMP transfer for that. And we'll also clarify the regulatory path to IND for this new modality. And in order to do this, we're asking for a raise of 15 million euros. And we intend to close this in the first quarter of next year. In order to summarize, I hope what I've shown you today is we've developed a revolutionary cancer therapy class with lower off-target effects for our patients, with new targets that are addressable for the first time, and it offers flexible programming for different diseases. And we intend to turn this into an effective treatment for different cancer patients, offering them a better quality of life and unlocking a huge platform potential in several multi-billion-dollar markets. If you're interested, want to learn some more, please feel free to contact us. Happy to take any questions, and thanks for your attention.